Please start.
Yeah, yes, just a minute. Okay, so let me just open the share my screen so that accordingly we can start.
Ma'am, hope you have received the documents whichever I have shared regarding the pathway analysis and also for the genes, the details of the genes.
Yes, so the document that you have shared is this one from July 8.
Your screen is not
Like
visible.
my screen, why is
I
it
can't
not
see anything.
visible for you also?
No, no, it's not visible. Do you have access to share screen? Can you please let me know that?
Just a minute. Like you are not allowed to share your
Yes.
screen.
Yeah.
So can you please provide me the access?
Yeah. Please try now.
Okay. Yeah, so the document that I saw from Vajanti is this one, the results DEGs in your classroom.
No, 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 no. You asked me to upload it in the drive.
Right, right. So in Drive it is, I don't found like your results actually in the code section. Okay, this one. So you have the cake pathways already filtered out. How many pathways you have filtered?
So I have highlighted all the pathways whichever I have put.
Okay, so we need to proceed with these pathways and the genes which are involved in these pathways. So we need to focus on them, right?
Yes.
Have you checked their gene expression like based on the sample types? Because no, based on the condition, like whether they are of your interest or not, their
Yes, the
biological
genes,
function.
yeah, yeah, whatever I have selected are of
Yes,
my interest, and I hope you have gone through the notes which I shared you because I was,
just a
yeah,
minute.
this,
Yeah, I checked it actually
you
earlier.
didn't
Let
check it
me.
before?
Yeah, yeah, I saw it, but
Uh,
because there was theory, no, so I was thinking that first need to discuss, then accordingly I can explain. So, where, as you have mentioned about all the genes and no proper evidence, so no evidence, or there are few papers it is mentioned, but they have not explained it. Proper
no,
means
like
what
uh,
exactly?
about CRC, like on colorectal cancer, they
Mhm.
don't have any specific role of that particular gene. That's what I
Okay,
have mentioned.
okay,
Apart
okay.
from that, they have
Mhm.
certain roles in their
Mhm.
discussion, like how they express in other biological functions, but
Mhm.
no proper evidence has been found in colorectal cancer. That is what I was meant to.
Okay, so this is a part of literature survey, right?
Yes.
Or any specific database you have searched?
No, it is
Okay,
completely literature
literature survey.
survey.
Okay, okay. So the genes for which no evidence is found, so that we can mention them as novel biomarkers
Okay.
or novel genes specific to colorectal cancer. And
Okay.
now we need to correlate them based on the network that we are making and the protein interaction network that we performed last time in the last class, so that we can say that if the gene, the FOS gene, is correlated or co-expressed with ENPP2 or not. Okay, whether it is in colorectal cancer or any other cancer. Okay, that
Okay.
will help you to like conclude something out of the new genes that you are getting, because already no theoretical information is given related to their biological activities in colorectal cancer. So what
Yes.
we will do is we will try to correlate them based on other type of cancer, so that we can mention that similar kind of their activities are expected or estimated in colorectal cancer as well. So they could be involved in the progression of colorectal cancer. Okay, so that's how we can correlate. And okay, so for most of the genes there is no such publication or data available. But okay, so for which genes are there, for which gene data is available, for them we can check first, because accordingly we can work with those genes, novel genes separately, and for which the evidences are available. No proper evidences, and for these two there are evidences for. Okay, so we will select actually those genes which are having evidences, or maybe we can also check for, because the genes for which information is not there, so that information is also not stored in different databases, right? That's why it is also not, you are not getting in different literatures. So that's why we are trying to skip those for which evidences are not there, and will work on the genes for which evidences are present, so that accordingly we can filter them.
So which one are we planning? Like in out of the 24 genes, let's say around 11 genes are they're having some evidence. Remaining 14 or 13 may not have evidence. So we are going to say that 13 genes are novel genes. And are we going to present any data on them? Like what is their role or what they're going to do in other cancers right now?
Yes, right. So in other cancer, we need to mention that for example, in lung cancer, or maybe in gastric cancer. So in gastric cancer, this particular gene is involved in any specific biological function, whatever we have identified during annotation or functional annotation. Then we can mention that in colorectal cancer, these genes are estimated to play this particular role, as they are involved in the other cancer for this specific role. Okay, so accordingly we can mention that, we will see later, but right now we are intending to see that in which particular, like today the session is about tumor microenvironment, right? So for that we are looking for what different tumor related cell types where your genes are most activated. So that's why I'm filtering out some of the information, because we will looking for each specific gene in the database that we have, or the tool that we have. But in that case, we need to filter the genes that are already existing in the literatures, because in databases those information will be stored which are published, not no data on CRC. Okay, so you have mentioned two terms, no proper evidence and no data on CRC. So how we will differentiate these two things? Can you please mention
Both
it
of them
in?
are same. I just mentioned like no proper evidences are present from humans colorectal cancer.
Okay, so there is improper evidence present, like any information, like in the colorectal cancer analysis, like the study that they have done, they have mentioned this particular gene in their study, but they have not discussed something like that. Is it
Yes,
that?
like for example, KATNA, like Katlani, like
Mm-hmm.
that gene, they said like it is indirectly related to colorectal cancer, but they haven't processed, like there is no proper data on their biological function has been presented. Like what is the
Okay,
actual role in cell process?
okay,
So
and
if you have
neither they have gave reference, any reference for other paper.
they have given the like they have simply mentioned like it has an indirect role. That's it. And that is why I have given you the entire chunk. If you have gone through this entire notes, you might have got some idea like what I have
Mm-hmm.
given on the data
Yeah,
on each
right.
and every gene.
So, yeah, so references I can see, but what I am asking is if they are mentioning that it is present in CRC, like in some way. So, they have not mentioned how, right? But just
Yes.
mention it is present in CRC.
Uh,
But the problem is where they are mentioning, just a minute, it's while not indirectly, directly implicated, it's possible that. So this is mentioned in the paper, right? The whole
Yes.
statement.
Yes.
Okay, that could play an indirect role in CRC through its involvement in cell division and migration. So they have mentioned this their biological activities and processes also relevant in cancer development, as mentioned in the specific paper as they have mentioned here. So you
Okay.
need to also go through this paper, because in this paper they might have discussed the role of CRC in this particular gene in CRC. So that is one extra thing that we should do if someone is saying, rather than mentioning that there is no data. That's why references are there, right? So that we can refer to that particular paper and see that how this
Actually, that
particular gene
paper
is developed.
talks about a different set of gene expression. Okay. They just gave a line stating that Kaklani has a role, is an indirect role in colorectal cancer. So that is how it is mentioned.
Okay. Okay, then what we will do is we'll try to, okay, let me take all the genes as of now, because anyway we are not having like certain information on their relativity with CRC, but for some genes it's certain, right? For GP, NMB, and for
Yeah.
other genes it is related to CRC.
Yes.
So what we can do is, just a minute, we need to check their presence first. Okay, so in your slide that you presented here, where is the slide?
The slide just is above on the results DEG.
You
In
have updated a slide.
yeah.
So there actually, what I had to mention is, just a minute, I am going through it because I just reviewed it like earlier. We have already discussed these things, right? So these figures you will be revising. And yeah, for survival analysis you can generate different figures using, I think I have discussed the tool which is Kalpan Mayer tool, or KM plotter we can say. So in this KM plotter we can make survival analysis figures that is more relatable. So here we have for specific for colon cancer, that is again the colorectal cancer, specifically originated from colon. Okay, so here you can add multiple genes that we already have and generate the uh, survival plot. So that, let me just complete this because it was incomplete in the last session. So where were you like all these days? It's been so long, the last class which I took.
I was occupied with some exams. That's why.
Okay, okay. Um, compute. So if you are not selecting, then it will automatically compute Spearman's. So we can select according to our, and let me do just analysis. Where is the option to submit? Okay, so plot options you can modify. Here are all the options given. What are the thickness and all you need. Okay, and here we have different information that you want, this recursion free survival or overall survival. Okay, so these you can select accordingly and you can see that how many number of samples are there, based on which it is giving the survival analysis report. Okay, and so it is considering all the stages for colorectal cancer. If we are working on early detection or primary stages, then accordingly we can select. So that is one thing, and then you can go for plotting. So it will generate survival plots for all your genes which you are interested to study. accordingly, if you are identifying some novel genes, as we have seen that no much information available, so for them only you can provide those survival plots in your paper, because the other genes which are already well known in colorectal cancer, then for them data must be available, so you don't need to provide for all 24, just provide for 11 or 13 whatever genes which are novel. Okay. And yeah, network analysis we already performed. So now, since you know that which are the novel genes for which you don't have any information or related to colorectal cancer, and the other genes for which we have information, so with that perspective what we can do is again perform the network analysis, and then see that how these genes are interacting with each other. So let me just quickly do that thing as well, because I have seen that you have added this network. Okay, it is the same one. Okay, here. So this we are updating with survival figures, because this is not too relatable for us right now. We will be checking for overall survival anytime. Okay, but when we are working with different stages of cancer, then we go for recursion free survival or disease free survival. But overall survival is taken in general. Okay, so this will help you to conclude based on the survival plots as well. Let me show you the survival plots first, so that accordingly I can mention you that which are more predictive kind of plot that we are having. So here we can see in the first plot for this gene TGFBR3. Okay, there is not much difference between the high expression. If the gene is expressed high or low, the survival is having survival probability does not have much difference, right? But when we see for the other genes, we can see somewhat the probability is changing if the gene expression is high or low. Okay, so if expression is high, it means the probability of survival is decreasing over time. Okay, time is on x scale, so that will help you to filter your genes based on the survival. So those genes which are having more difference here, uh, those genes we call them as predictive genes. Okay, the that can help us to identify or predict the disease stage, that how with the time the disease will progress. Okay, so this is also not predictive kind of gene, that is FAM. So you need to check in your list actually, like every time you are checking, so you can download these PDFs for the survival plots, and you can check that like keep all the survival plots for the novel gene on one slide, and for the other genes which are known on another slide, so that accordingly you can compare and filter out the most predictive genes which will be important to study for your type of analysis or for any kind of analysis. Most of the time we focus on predictive genes that can predict the stage or the progression of the disease. Here also there is not a lot of difference here, because as we see the probability is in zero point 0.6 to varying from 0.7 to 0.8, so not much difference. But still, if there is, you are not getting too many genes as good predictive genes, then you can consider this as well, because somewhat there is a difference. Accordingly you can check for each plot, and you can see. So this figure is not related which basically, because this is hazard ratio and p value. So this is just telling that how your survival plot is being calculated or expressed here. So accordingly you can download all and check, so that you can filter out only those genes. So now you can understand, we are narrowing down our results, okay, so that we can focus on a small set of genes
I
to
guess I
discuss
have
in paper.
already done this
Just
survival
a minute.
analysis for
So
the
it's not in
for
your
disease
slides.
based survival.
So for survival analysis, only this slide is there in your paper, like in your PPT, right? I'm unable to see it here. So,
But I
okay.
have
So,
done that already. I can also share that too. I guess in last class
yeah.
I was sharing the screen and you were teaching me the same, and you were asking me where are the gene names. I told you it is inside the box itself. I
Okay.
remember that.
Yeah. So that's why I'm saying, no, you, if you update on slide, then only I will know that how much we have covered and what. So I'm repeating the same thing, right? So did you filter the predictive genes out of this? Because I can see not many genes are predictive. We have only few genes here, so maybe three or five genes. So those genes we can further proceed to check that how like how they are involved in the tumor microenvironment. Okay, so for tumor microenvironment or studying tumor microenvironment, first we need to understand about the tumor microenvironment. Let me just discuss about that first. So here if we see that you already know what is tumor, and since there are different type of cells working together to like progress in tumor or to enlarge it or to grow it larger, so we can see that what are the different modules or components of tumor we have. Like they can be from the tumor cells specifically or dendritic cells. So different type of cells are there. They work together. Some of them work in suppressing the tumor, and some of them are working in progressing the tumor. Okay, accordingly we can filter them based on their groups. Okay, so how many of them like which of them work in the support of cancer progression, and which are working in the against of tumor progression. Okay, accordingly here we have anti-tumor microenvironment that will suppress the tumor. Okay, that will involve apoptosis a process and cytokines. Then we have T cells that will activate the CD8 cells or CD8 plus type T cells. Okay, that will help in suppressing the tumor. But we also have tumor promoting microenvironment. Both includes in tumor microenvironment. Okay, so you need to understand that which type of environment specifically you want to categorize your genes in, or you want to study their function specifically in the tumor microenvironment. Okay, so all the modules of tumor microenvironment does not belong to tumor promoting. Okay, some are also involved in suppressing. Okay, so here we have one paper, friend or foe. I'm not sure if you have checked or not. This is about the microenvironment in colorectal cancer, and here they have mentioned different types of cells and different stages of colorectal cancer, and how different cell types are involved in these particular stages. So as the stages increases, the quantity of the microenvironment cells or the tumor promoting cells are increasing. Okay, so here we can see in this particular figure, when we have normal mucosa in colon, then we have normal cells, or we are also having precancerous cells, so that are in normal stage but not like malignant form, in not in malignant form. Okay, but once it is promoting to the advanced stage, or just initializing the cancer, or maybe reaching to stage 1, stage 2, stage 3, stage 4, we can see here the triangular boxes for green color, that is showing that there are some of the cells which are not dangerous for your colon, right? So that's why it is kept in normal stage. But as it increases, we have different type of for example, macrophages get into action. Then we have lymphoid cells. For example, we have T cells, that is CD4 plus. Okay, so that will help in promoting the cancer to the next stage. But what we saw in the last figure is, okay, where is the last figure where we have T cell, but CD8 plus type T cell. So that is helping in suppressing the cancer. So accordingly, first we need to understand that what are the function of each type of these cells, because all of these cells are involved in tumor microenvironment. But we need to know that which cell we are interested in, whether we are studying about tumor progression or tumor suppression. Accordingly, we will think about, if someone is saying, or maybe you are checking your gene activity or in T cells, then T cells are further categorized into CD4 plus, CD8 plus. So in that case, you need to check that whether CD4 plus supports the progression or it do the suppression of cancer. Accordingly, we can explain better in paper that your gene is enriched in or gene has enriched with the with this type of cell which are helping in progressing the cancer. Even if your data set is not from progression, it is still in stage 1 or maybe stage 2 cancer, right? That's why we are studying this study. So in that case, we need to first understand the function of each type of cells, and accordingly we will filter that which cells are involved in promoting the cancer. Then accordingly we can understand that how they are helping or promoting the cancer or the colorectal cancer, or how they will promote the colorectal cancer. So for that we have the tool here. Okay, so here we have the tool that is specifically working on immune kind of tumor cells. Okay, so immune related or immunity related tumor cells. Okay, based on, so specific to cell type, that will vary. Okay, so in which category your cells are lying. So based on, like as we just saw, suppression or progression. Okay, so this will provide you information based on single cell data, like widely used 190 data sets total were used, and out of them around 6 to 97320 cells were estimated or calculated from them, or their expressions are captured from 190 data sets based which are single cell based data or single cell based RNA data. So for each cell we are having enough number of, for each from each data we are having enough number of cells to quantify or to provide a generalized result. Okay, generalized result means, it does not mean, for example, in your study one gene is showing expression, upregulated expression, so you cannot declare it as a, for example, if you are working on just 5 healthy samples and 5 disease samples, which is very low number of samples, right? So we cannot provide a generalized result based on that type of data, which is very data set which is having very less number of samples. But if we are working on 1,000 number of samples, then in that case we can claim to providing whatever result we are providing is a generalized kind of results, can be considered for any kind of colorectal cancer. Okay, so here this stitch data, and this is for version 2. So here we have all type of diseases here, and we have for from colorectal also. So out of all the 190 data sets, the colorectal cancer information is from 11 total 11 data sets. Okay, so based on that, based on our gene of interest, we can search and then see that in which tumor microenvironment they are involved. Okay, so one by one, like as I mentioned, that we need to narrow down our gene list based on what genes are novel or what are most of our interest. Okay, as I mentioned that the novel genes that we are mentioning here, maybe for them, as we saw that for colorectal cancer there seems novel, there is no information. So you can try few here, but if the information is not there, it means since the gene is new for colorectal cancer, no studies has been published, that's why data is not there. But the for the other gene for which data is there, we can search for them. Okay, so let me just search for one gene, at least for trial. Any so that's why we have all 24 genes, so we need to do the same task 24 times. That's why we are trying to reduce the size of our gene list. And we can search for the, we can also download the whole information, whole data set information, and map with our gene list. That will also work, but that is an extra step, so we can just skip to checking for each gene specifically. Okay, here we have the gene name, and now we need to enter more information. So maybe the gene is already involved in breast cancer, so we need to remove that, and we need to give colorectal cancer. And cell types we are not mentioning because we are interested in all type of cells, as they are all belongs to a tumor microenvironment. Okay, but still we need to differentiate or exclude those cell types which are tumor suppressing cell types, or even promoting cell types, depending on the objective of the study. That's totally up to us. We can also select multiple cell types. For example, we just saw that the CD8 T cell is working in suppressing of cancer. Okay, but we are interested in promoting of cancer, or how which cells are supporting the progression. So we will try to remove that in the list. Okay, and if you are not selecting any, then it will consider all whatever list is given. So let's just move. And why all lineage? Because we are interested cell derived from any of the group, either it is from myeloid or an enthelial cells, enthelial tissues, or fibroblast. Okay, so we are trying to get from all, and the data sets we can see here. You can also check for these data sets in geo database, that what are those data sets, how what are they are specifically for colorectal cancer or not. So this is from clonal expansion. Okay, and of specifically for T type lymphocytes associated with tumor infiltration. Okay, so this way we try to understand more about our data. Okay, so and now we can search for this, so that it will help you to get the result and understand the uh, correlation of your gene better. Let me just filter this out because we are not getting results in the table right now due to limitation of cell types maybe. So let me just remove this as of now. So this gene was novel or not, I'm not sure. CYBRD1. This is not novel, so the information should be there. Okay, so it means this data set is not uploaded or not updated for this particular in this particular gene. So we need to check for other genes also. If you are getting, it's fine. If not, then we will check in other databases which are related to tumor microenvironment, so that it can map our gene to the specific type of cell types where they are active. Okay, so now what we are doing is, what we have done is, we need to check the cell type annotation. So earlier it was about major lineage and minor lineage, like majorly if it is associated, but right now we mention malignancy, that is for tumor, and it is mapping our gene with different types of data sets in different type, from different type of data sets. The information is deriving from that within immune cells your gene is active, and within different malignant cells, like wherever a tumor is there, it will be active, and then stromal cells. Okay, so all these cell types will be considered within tumor microenvironments. And here we can see within this particular data set, which is from SmartSec2, where we are having more score log TPM value. So transcript per million. Okay, so more number of transcripts are present in this particular study, where your gene has more involvement in more number of stromal cells, or more number of stromal cells are present. Okay, so we can say that highly significant stromal cells are highly associated, or gene is highly associated with stromal cells type microenvironment. Okay, accordingly we can check for all other genes, because this database is providing uh, can we add more? No, we can search for one gene at one time. So whatever gene is available, the information is available, you can download the information, and then accordingly, either you can make the table with this information, whatever information you are getting, or you can just directly download the figures, okay, directly from here. Or you can also see the gene correlation here, like with your gene, what other genes are there with which the correlation is there. And you can also search the correlation with the gene of your interest maybe. So you can just just filter it out or see. So once we download this particular information, we can also check for a specific type of gene that we are more interested in. Okay, so we can select the gene, for example, it's if it's in our list or not. Let's say TGFBR3. So if we check for the gene, then we can see that if there is any relation, or then cell type is present or not. Just a minute. TGFBR3. Okay, so we got these data sets have this particular relation mentioned between these two genes. And TGBR3, you need to check in your table that whether this is a novel gene or already known gene. So this is already known gene.
It's
And
not novel. It's already
yeah,
known.
yeah, it's already known. So that's how we need to now focus on the genes. This is important also, that if we are correlating two genes which are already existing separately, right, information is there in papers but separately for independent genes, but we can also correlate them, the information that we are getting. And apart from that, we can also work on their relativity with the novel genes. Okay, so let me check for any novel gene here. MP1 JP, or maybe BCHE. No, since the gene is novel and we are working specifically on colorectal cancer, that's why no information is available. Okay, so for that, what we will do is we will focus on network that we are going to make. If the two genes are correlated, for example, the already known gene, that's why I'm asking to filter the list of genes which are already known and for which no evidence is present. Then accordingly, we can map the two lists between the genes and then study their relation. So let me just focus on BCHE and the one CYBRD1. So that's how we will try to make the network. So when we have objective in mind that what we want to focus on, then in that case we can do many modifications in the network. Okay, so now we have network here with just two genes connected. Here we can see. So this gene where we are interested more, and the other one is this one, which are not connected right now in this state. But now we have settings option where we can modify the settings to know that any kind of connection present between these two genes, because we know that one gene has is highly active in different types of tumor microenvironment and different types of cell, specifically the cell types now we also know from within the microenvironment. And for this gene we don't have any information. So if the expression of this change is affecting or changing, then if they are correlated, then this gene might have probability to get affected with the gene if they are co-expressed together or like locally present. That's why we do gene annotation, that to get the cellular component of each gene. If you know that they both lie in the same cellular component in the same section, it means they both will be like the other gene will be definitely affected by this gene because they are existing in the same component and they will be having some relativity. So within a string we will try to modify first. Let's just take the confidence level to the minimum we can. So if it is not bad if we are going for low confidence, and there is also option to customize the value because we can go even lower than this. Okay, just to understand that how they are related to each other. Okay, so now we will go. It's just that if you are reducing the confidence, it means the species that you have selected. For example, Homo sapien. So in that case, it may give result from other species as well. If in Homo sapiens, the information is not present. Okay, that is just the difference. And so let's just try for submission. So we'll update the network. And now we got the connection between our genes. So these two genes are totally not interacting. So we'll keep them aside. And we have the gene of interest, this one. And where is the other gene? CB BCHE. Okay, so this BCHE gene is actually interacting with FOS, that is a FOS gene, but not with CBRD1. Okay, so now what we can do is either we can get more genes from the outer layer that are not in your list, but they are interacting with these genes. Okay, so we can take 5 extra genes in our list so that if we can see that if our gene of interest are interacting with each other or not. Even if that is not possible, then what we will do, we will not focus on this particular gene with its interaction with BCHE. Maybe they are no way interacting. Okay, so without like delaying or taking more time and unnecessarily, we will focus on the other genes which can be related. Like we can see now in our network, other genes are all connected except the 4 genes that we are having here. Okay, so we will focus on the other genes. So in your list, you can check on the any other gene which is known, maybe TGBR3 that is here, and we can see that how it is connected with BCH. Okay, so now we need to study this particular connection. And where is TG? Yeah, here we have TGBR3. Okay, so if the gene expression of TGBR3 is affected, it can affect the expression of FOS, and FOS will affect the expression of BCHE. So we can try to establish this particular relation by showing this network. And when you click on the edges, you will be seeing this information, that how and where in which particular organism this particular information is provided, that how they are like considered together, connected. Okay, in what terms. And there is a public PubMed abstract given, so you can also go to that paper and see that how those genes are mentioned. Okay, so accordingly we will further study. So this will take some time to study the genes together. Accordingly we can conclude our result based on the interactions that we are getting. Okay, and yeah, so no interactors we got, but let's see if we are interested in CYB only in case. So that there will not be case, but in case if you are interested in future, so for that case we take some extra genes which are anyway connecting our CYB gene with our BCHE gene, okay, where in which we are interested. Okay, why? For any specific reason we can be interested, as it is a novel gene and we want to study its function or its properties, then we now we have its connection with FOS, so we can also study it with respect to FOS. We can study it with the CLU gene, that is the external gene in our list. Then we also have A2M gene with which it is interacting. Okay, so based on these interaction details we can write more about it. Like you can see here, the co-expression level is higher here, 0.08, like considerably co-expression is present. And then we can further see the reference for this co-expression. So these two genes are co-expressed together. Okay, so expressing together means if the expression of BCHE is changing, then definitely the expression of other gene will be changing. Okay, so that's how one gene impact other gene, and that is the significance of studying the network. Okay, so hope this is clear, and accordingly you can filter out the genes and study them based on the gene of interest and their interaction that we are identifying here, and the cell type. This table you can, maybe if there is a download, yeah, there is a download option, the correlation download option, so you can
Okay, ma'am,
download so
I
that.
have a doubt. Is there
Yes.
any other software or any other tool to discuss about the tumor microenvironment, or just this one alone?
No, no, there are other tools also. For example, estimate tool is there. So here also this one is estimate from MD Anderson Cancer Center. So here what happened is basically it is not specific to gene. Okay, it will give you from all the data set that are available in this particular database. It will give you the list of the cell types that are involved in the colorectal cancer. Okay, so not specific to the gene type but the disease type. Okay, so that's why this is better if you are going with the gene type, as we are focusing more on DEGs. That's why it will be more significant for us. Apart from that, there are also other databases, for example Excel, then timer database is there, timer DB is there, but they all are somehow not too good, uh, more than this. So you can check, because okay, so one more thing is about this is that it is specific for the immune related tumor cells. Okay, so for that, for the other type of related genes, we can check in other databases like timer DB and other databases that I mentioned. Else, we can also check in different publications while doing literature survey, that where these particular genes are involved, whether they are involved in B cells or T cells, and accordingly we can filter them out. So we need to do some tasks, like take some time to like get all this information, make them in a table. Accordingly, we can finalize our conclusion for the paper. That will help. So, and also for functional annotation, I have provided you the script. And let me just see, this is for bar plot. I have provided you the script. What information is there? Yeah, this script I have provided, and we have already discussed, right? This script of to make the like gene ontology term. So if you are mentioning all, it will generate for all. And so I was trying to generate the figure, actually error in bar plot. Height must be vector or a matrix.
And your voice is breaking actually, it's not clear.
Yeah, so what is it clear now? Hello, is
Yes.
it clear? Yeah, so what
Okay.
I'm saying is this script I have shared, you can generate the figures, as we have already generated figures during performing the analysis, I guess. I think in analysis we generate the figures, right? I already have those figures which we generated for cake pathways and all the stuffs. These are the figure like the geoplot. You have that in PPT, right?
This is for specific RNA-seq and microarray data.
Yes,
Is it
right.
for, I don't want for the microarray data or for the DNA-seq, RNA-seq data. I want it for the 24 DEG. For that, we need to create the gene ontology.
Yeah, so that's what I'm mentioning is that you have to use the same script which I have already shared also, like the functional annotation underscore all. This script will help you to generate the figure using the file that I will upload. Here we are using the file 24DEG's FN expression matrix. This file you can use where we have the list of genes, this one, the list of genes here, and we just need the list of genes to generate the gene ontology. Okay, so it will generate the all the geo and all the maps that we need for bar plot. Okay, so this is not generated actually. I'm getting some error. Okay, so I will check that maybe due to some package or what information it is giving me error. I have saved the result in 24 DEGs. I will upload this file as well so that you can use the same, and based on this you can execute the code and get the same figures that we have generated for all the ontologies we have already performed. Okay, so this type of figure you need, right?
Yes, yes.
So yeah, we can generate that using the file and the code that I have already uploaded in the drive today. Okay, so that will help you. And regarding the tumor microenvironment, need to do the task specifically based on the genes which are novel and not novel, and and also first filter your important genes which are involved in these pathways. So with these genes, proceed for checking that which are novel genes, which are not novel genes, and accordingly you can go for tumor microenvironment check and just filter out the type of cells which are involved and make the table for that. Accordingly, we can write some conclusion based on further literature survey that if there is some information present to connect the information that we are gathering.
Is that all about your microenvironment?
Yeah, so that we just need to perform this particular analysis, filter out the cell types that we need, that we are interested in, or we want to study, and that is like all. We just need to now correlate. Once we are getting all this information, maybe the other gene also, not just these two genes. The other genes are also having the CD, are involved in with CD4 T cells or myeloid cell, fibroblast cells. So then we can mention in results collectively for those genes which are enriched with these type of cells and they are actively involved in colorectal cancer. Okay, and we can cite the tool as well so that accordingly it will be mentioning that what type of tumor microenvironment cells that we are having. So that task need to be done. So
That you
if
will be using the David software for the correlation, for the analysis.
no, David tool we use for functional annotation. Functional annotation we did actually. Did I mention functional annotation? Actually, we did with here you have added for
Ma'am,
enricher.
actually, I last class itself, I told you that I'm not satisfied with this enrich R and I want to do with David.
Okay, so let's go with David. So David, we can open. This is the David annotation tool, okay? And you can start your analysis from here. Here they have made like updated their interface actually, so you directly get options of functional annotation or ID conversion. So we can perform functional annotation here, and we can paste the list of genes here in this particular section. So let's proceed for that. Okay. Once the list is pasted, you need to mention the step 2, which is what you have pasted. So we have pasted actually the official gene symbols. Okay. These are the official gene symbols, and we need to mention the species, which is Homo sapiens. And then we can mention that it is a gene list and submit for functional annotation. Okay, so this is the okay. This is the result from functional annotation. You can see that all the 24 genes that you have given as an input are here or mapped to your David result. Okay, so and then gene ontology you can check here. For gene and ontology term, we need to check for biological process, the direct results, cellular component direct results, and molecular functions direct results. The same results we were also getting from the script. I shared you the R script where we are doing functional annotation, and for the same thing, we are getting the results. So these results which we got actually are not from enrichR. The same thing we are doing now in David. You just need to download the tabular data and generate these figures. We already have used the tool and generated these figures. So you don't need to go again and do David or annotation again because you already have biological process, cellular component and all. But still, if you want to see that how to do functional annotation with David, so let's proceed for that. And first we will check for biological processes where your genes are involved. You can see total 21 genes are identified involved in the biological process reported in David. So you can check on chart, and within chart you are having these information that all the biological processes where genes are involved in the first one, which is TGFR, that is growth factor actually for beta receptor signaling pathway, and where your three genes are involved. Based on the p-value, you need to sort your genes accordingly, or the your terms accordingly. It will help. Okay, so we can download this table. Okay, here we are getting this type of results. So you can save it, Control S, and it will save your text data here. You can just rename it BP for 24 genes. Okay, you have got the table here downloaded. Similarly, you can also download the other results. Okay, where is David? Just a minute. Where I was doing the just a minute, let me check my tab. Yeah, this is that. And then similarly, you can download cellular components, because you know cellular components are also important, as we just saw that we need to know if two genes are lying within the same cellular component, then their activity, their co-expression values will be having more like consideration if they are in the same cellular component. So accordingly, we try to discuss that in papers when we are publishing or writing about them. So we have 13 genes involved in or present in plasma membrane. Okay, and 11 genes in membrane. So membrane and plasma membrane are kind of similar only because that is plasma membrane is one module of membrane. Okay, so there will not be different genes here. And then three number of genes in cell cortex accordingly. If there, if you need more information here, because very limited information is present, or maybe for biological process or any type of thing, so we have options here. On the top, you can see there is an option tab. When you click on plus, we have options that minimum number of genes in my list could be one. Just give me the name of the cellular component wherever my genes are there. Okay, so we will reduce the number to one so that we can get more genes, and then rerun using the options. So once we rerun, we will get more number of lists, maybe extra value we will get. And even if by changing just count one, we are not getting more, then we have more options in this particular section where we can increase or reduce the ease values. Maybe here that if it is 0.1, that is the score that we are getting for significant number of cellular components, we can increase it to 1 and then check that how many results we are getting, and accordingly we will get more results. But depending on our requirement and how much gene there are involved, now in cytosol you can see we have 8 number of genes involved, so kind of considerable cellular component that we can get here, right? So that's why accordingly we modify the options if required, and we will download the file. We have the text file you can see on the bar, so you can directly control S to the web and it will save. So cellular component for 24 genes. And later you can open these tables in Excel to see the data in tabular format. Similarly, molecular functions we have. So molecular functions we will download from the chart. Okay, so you can do that later. One thing I need to mention is here we have pathways option. Okay, and pathways, KEGG pathways are more important or more related to disease kind of a study. So we will focus on KEGG pathway and download the chart. Out of 24 genes, only 11 genes are identified to be found in any reported KEGG pathways. So we can open that and see that how they are involved or what pathways we have involved, whether they are same that we got from the script, because there we used KEGG pathway in the script, the code that we use to generate the pathway analysis or all, then we used KEGG pathways. Here you can see KEGG pathways. So enrichment of genes in KEGG pathways, okay. And also I think we also use reactome pathways. So for reactome pathways also, we got the results. For KEGG pathways also, we got the results. So no result will be different from here, the tabular data that we are generating from here. So that is totally up to us that what approach we want to go for annotation or any kind of a study. Okay. Here we are getting very less number of pathways. So again, we can modify the options to 1, because if we are mentioning 0, then it means no gene is involved. So that is not relatable to us. And then ease value we can increase so that we can get more number of terms here. And now we get more number of pathways, and the same pathways that you got in your result here in the table, right? Oxytocin signaling pathway and everything is there in the David. So that is how we perform analysis with David. You just need to get these tables. So once we have downloaded, you can just open them in Excel. BP BP we downloaded, but okay. So in which folder it was downloading? Just a minute. It's in download CC and BP. Okay, just I will show you quickly this one. and then we can wrap up so that you can work on the filtering of genes and then getting their tumor microenvironment so that further with literature survey we can understand that what activity they are involved in
Yeah.
those particular
Like,
cells.
yes, I have it up. Like for those genes which are very novel, like they are
Mm-hmm.
not being presented for any type of cancer.
Right.
So for those, what can I do? Like I don't think so. In some databases, we don't find any data on that particular genes, right? For example, if it is TGFR, and I don't find any relevant data for the tumor microenvironment. So what can I do for that?
So for that, what I'm explaining earlier was also that first we have to mention, like whenever you are writing the paper, you need to mention that this particular gene is involved in the specific biological process or the molecular function which is specifically mentioned for that particular gene in different cancer. Okay, also, or you can also check for multiple type of cancers. So in different cancers, what role it is playing, and then further we can mention it as that there is like it is estimated that it will be like playing the similar role in colorectal cancer as well and progressing the colorectal cancer. Okay, so that we can write for those set of genes which are totally novel. The other step we can also do is to correlate them with the already known gene in colorectal cancer through network analysis, as we just saw, that correlate that how they are interacting with your known genes which have well established study in colorectal cancer. Okay, so in that way we can say that this gene are differentially expressed. It means their expression are changing. So if the gene expression of one gene is changing or the known gene is changing, then it will modify or it could modify or alter the gene expression of the unknown gene, and that's how it may be playing role in colorectal cancer as it is playing role in different type of cancers reported in studies. Why I'm not able to see my files? Okay, just a minute. File type I had to select. So is it clear what I'm saying, like
As for
how
now,
you
it
can
is clear.
report that?
Yeah. So yeah, as for now, it is clear. So I will just check for
Okay.
the biological process, and I will
Yeah.
just go for the correlation with the known genes to the same
Hmm.
string database. Then I can write the paper accordingly, like the differential expression of the known genes may affect the expression of the unknown. And yeah, I guess only with this we can report these things. Yes, ma'am. And
Yes.
for next class, I'd like to go for the primer designing for the 24 genes.
Yeah, for next class we can perform primer designing. And do you have a specific overrepresented sequences in the gene, or we are directly going with the whole sequence of the gene?
Sorry, come again. I don't get it properly.
Like, do you have a specific set of overrepresented sequences of your gene list, or we are going with the whole sequence of the genes?
What represented means I'm not sure about that.
Um, Okay, so let's discuss that in the primer section only, because in that case, if there are not already defined overrepresented sequences, then what we will do, we'll first define them, and then or first identifying them within the gene sequence, and then we'll go for primer designing. Okay, that we can do later, because today itself we are doing a lot of things. So, okay. So now what I'm doing is trying to import the David data.
Actually,
And
in next class, that would be my last class. So in the last class, I want to get the primer sequence for the 24 genes. And
mm,
after the next class, I might take one more class where there will be discussion of my paper. And I would like to wrap up this.
okay.
And may
Mm,
please also, I would like to know how much you will be charging for primer designing As you said, the software is chargeable. I'm not sure about that.
Yeah, software is licensed actually. So I think okay, we haven't discussed that yet. So maybe we'll tell you after this class. Give me 30 minutes after the class, then accordingly can inform you. Okay, and then here we have the text data that we have downloaded from David. You can perform next and then give the delimiter that you want to separate your data from. It is already delimited with tab. Then you can go to next and finish so that you will get tabular information for your data. Okay, this is our biological terms and these are your gene list. Okay, too loud color. So accordingly, you will get these results and whatever you want because well, like with scripts also we have done the same thing. If you are getting any more more information here or more terms here, then we can also use this to generate similar kind of figure. Otherwise, with script also it is doing totally fine and giving all the terms. Okay, so hope that will help you. And yeah, so let's wrap up the session. You try to work on the things that we have discussed today. First, generate the survivor plots, add it in your PPT so that it will be updated, and then do on work on the things that are novel and non-novel genes accordingly. Try to make the results and add in the PPT. And if you have any kind of query, you can mention it to the team. I think Bhavani number is there. You have your WhatsApp number.
Sorry?
You have the WhatsApp number of our team, like Bhavani's number.
Yeah, yeah.
Okay, so you can directly ask the query there, and I will respond you. If you are mentioning your query in the classroom, then I will also check in your classroom, but you need to mention in messages so that accordingly I can understand. Okay.
Every time I do that.
Yeah, so do it regularly. Otherwise, we are losing track already. It's been so long, and now we are like, okay, it's almost going to be end. So now it's no problem. But still, if it was regular, so totally fine, so that we can be on track and completed it much sooner. Okay, so hope that will work. So primer designing is left only. If there is any query from this particular session, you can drop in a classroom and mention it message. I will answer it back to you. Okay.